I'm very happy to introduce one of my colleagues at Mayo Clinic, Dr. Rafael Jimenez, who is very much interested in the history of pathology, and I'm very happy that he accepted to talk with us, although his title is quite a mouthful. So the history of Angiostrongylus, Costaricensis. So I have a feeling it's related to Costa Rica, maybe, since you're from Costa Rica. How a pathology and parasitology collaboration expanded the differential diagnosis of acute appendicitis. So I'm really looking forward to this talk. So Rafael.
Thank you, and thank you for the organizers for inviting me today. It's really an honor to present at this meeting. So yeah, quite a few of words in that title, but let's start with it. And yes, indeed, it has to do a little bit with Costa Rica. And this is an entity that was described there in the 1960s. So what was Costa Rica in the 1960s? There had been a small but significant armed revolution in the 1940s, and contrary to most armed revolutions, this one actually ended up with a very liberal constitution for the country. So by the 1960s, Costa Rica had already been about 10, 15 years with this new system and had actually resulted in quite a bit of political stability. One of the big visions that came to concrete after this was the abolishment of the army, a characteristic that is still in place and is one of the few countries that has an abolishment of an army. It was just dwelling into industrial economy, but for the most part, the economy was really agriculturally based. One of the big triggers of that armed revolution in the 1940s had been the installation of a social security system, which actually survived the revolution and was consolidating as the healthcare model that the country would use for up to this date. Still, the healthcare was quite limited, especially in rural areas, and tropical parasitosis were still endemic. As far as pathology, there were only 2 pathology services in the country, in the capital city. So every pathology specimen was transported to one of these 2 laboratories. So at that time, clinicians, both surgeons and gastroenterologists, started noting this repeated presentation of predominantly preschool and school-age children with some younger adults that presented with a picture that was clinically indistinguishable from acute appendicitis. These patients presented with a right lower quadrant abdominal pain, fever, anorexia. In some of them, physical examination would reveal a tumor-like mass or a painful rectal exam. And laboratory-wise, they had leukocytosis with eosinophilia. Intraoperatively, the surgeons would find these erythematous areas, and they were surprised that despite the clinical presentation, the findings were not circumscribed to the appendix and actually included changes in the terminal ileum, the cecum, and the entire organs in the right lower quadrant. So enter Dr. Rodolfo Céspedes here. He is probably the father of modern pathology in Costa Rica. He graduated, he actually went outside the country and studied medicine in the University of Chile in the 1940s, came back, and then got a second scholarship to study pathology. So he went back to Chile to train as a pathologist there. He, upon his return, the University of Costa Rica was founded, so he became actually the first professor of pathology of the University of Costa Rica. He was a scientific, inquisitive mind and was constantly pushing academics in a third world country. He was a founder of the Acta Medica Costaricensis, which is Costa Rica's first medical journal. He received these cases and started noticing, again, that the process was not confined to the appendix. Actually, in some cases, appendix wasn't even involved. And he would find these areas of stenosis in the terminal ileum or in the ascending colon with thickening of the bowel wall that you can see here. Additionally, mesenteric nodes seem to be quite enlarged and concerning for malignancy. But when he would take a look at these specimens in the microscope, what he would see would be that instead of the typical neutrophilic inflammatory infiltrate of acute appendicitis, what he would see is a eosinophilic predominant infiltrate, plus minus some giant cells with poorly formed granulomas. Sometimes the process would be associated with transmural necrosis and transmural necrosis, and not a lot of process really center in the appendix in many of these cases. It was not until several years that with extensive sampling, he was able to find parasitic forms within the mucosa of the colon. And some of them were egg forms, like this one's here. Some of them were larvae. And he noticed that many of them tended to be present within the mesenteric vessels of the ileum, appendix, and ascending colon. So obviously at this point, he knew that he was dealing with a parasitic infection. And so he called one of the few microbiologists that were in the country at that time, whose name was Dr. Pedro Morera. He was an helminthologist. He actually had left the country and studied in Pavia, Italy, and in Tulane University in Louisiana, which was pretty uncommon in the 1950s, 1960s to have this resume. He came back to Costa Rica, and he actually was named professor in 3 schools of the University of Costa Rica: the School of Medicine, the School of Biology, and the School of Microbiology. And his daytime job was a microbiologist at San Juan de Dios Hospital, which is the hospital where Dr. Céspedes was working. Now, obviously, he got quite interested in this, and they started studying and accumulating cases with these findings. So in 1967, Dr. Céspedes and his residents, listed here, and I would actually do a parenthesis and say that all these residents became eventually chair of pathology departments in Costa Rica. So underscoring the role that Dr. Céspedes had as a father of pathology. And Dr. Morera, they reported in the Acta Medica Costaricensis, they reported 31 cases of this entity. And notice that in the more than half of these cases, the clinical diagnosis was acute appendicitis. These cases were collected over a period of a decade and a half, more or less. They all had cases, they excluded those cases where they couldn't find the larvae or the egg, so they all had parasitic forms. A few of them had thrombosis and infarct of the bowel wall, and that included 2 patients that actually died of perforation and peritonitis. They knew at that point, 1967, that they were dealing with a unique parasite, and they hypothesized that it could belong to the genus Strongylata, but they were not able to further characterize it. So it was not until 1971 when the 2 of them, Céspedes and Morera, were able to microdissect these parasitic forms from the paraffin blocks, or in fresh tissue, they were able to digest the tissue and made a very meticulous description of the morphology of the parasite. And they thought that it was very similar to Angiostrongylus cantonensis, one of the other Angiostrongylus species that is pathogenic to man. But they thought that it had significant difference that would warrant its classification as a separate species based on the morphologic findings that are listed here, and that Dr. Morera meticulously annotated as drawings in his study. And obviously, these morphologic features were corroborated many years throughout, and this is just one example. This is an excellent paper of a group in Brazil who actually described the morphologic findings of the parasite with the esophagus there, the intestine. This is a testis in a male form. This is a female form with a ovary, and then the terminal portion of the female form with a vulva and an anus, and of the male form with a copulatory bursa. That is much better appreciated here in scanning microscopy, and you can see the presence of this lateral ally, which is quite characteristic and definitory of the species. Here is the oral cavity or oral orifice. These ridges apparently are quite important in distinguishing from other species. Here's a female form with a vulva, an anus, and here you can actually see eggs being exiting from the vulva right there. And this is the copulatory bursa of the male with two large spiculae that are used for copulating with the female. So this became, of course, Dr. Morera's obsession. And he, in a very detectivistic approach to this problem, he knew that the only way that he was going to be able to decipher what this parasite really was to finding the host. So he went to the houses of the patients and started putting traps all around the house and capture every animal that would fall in those traps, dissect them, and try to find the larvae. And finally, he struck luck and find the larva morphologically identical in a rat that's called Sigmorum hispidus. And once he knew that he was very likely then dealing with an Angiostrongylus species, and Angiostrongylus species are known that their intermediate host has to be a mollusk. So the next stage in this second paper was finding the mollusk around the houses of the patients, and he eventually was able to find the larvae forms within the mollusks. So in 1973, he actually published this third paper in which he inoculated both the artificially inoculated the definitive host as well as the intermediary host, described the timings in which the larvae migrated from each organ within the host, and came up with the life cycle of the parasite. So the life cycle, let's start here, starts with the rat eating the larvae forms. Within the rat, the larvae migrate to the lymphatic channels and then eventually enter the general circulation at the level of the lung. And for whatever reason, they tend to concentrate into the tissues in the right lower quadrant of the abdomen, meaning the mesentery, the cecum, ascending colon, terminal ileum, and appendix. Within those sites, the larvae mature into adult forms, and after copulating, they procreate. They lay eggs in the mucosa, which when they hatch, they exit the mucosa of the bowel in the rat. Then the mollusk will eat the larvae, and within the mollusk, the larvae would migrate from the intestine to the tegumentary system of the mollusk, and eventually to the mucoid glands, where they actually are going to be excreted in the forms that are needed for the mice to continue the cycle. So the human is actually an accidental host, and when the human consumes the larvae, it's gonna is gonna recreate the cycle that happens in the mouse with one exception, and that is the larvae are not able to exit through the mucosa of the bowel again and to infect other, and thus to propagate infectious forms. So human-to-human contact or dissemination is not possible because of this phenomenon. In 1980, the group of surgeons from the National Children's Hospital in Costa Rica reported their first experience with this entity. They included 116 cases gathered within a decade. And look at the, over here, you can see the presentation symptoms. You can see that most of these patients would consult because of symptoms really suggesting of acute appendicitis. This is more or less a distribution of where they found the findings. They were, one interesting fact is that they were able to correlate a peak incidence of this process with the rainy season in Costa Rica, supporting the life cycle that Dr. Morera had described. And also they reported on co-infection of a portion of the patients with other parasites. Dr. Morera continued his entire life dedicated to this issue. He published more than 20 papers on Angiostrongylus costaricensis, all the way to his last publication in 2008, where he described the immunodiagnosis using an ELISA assay. He died in 2012, and upon his death, he had already achieved a lot of laureates, and he was named advisor to the WHO. He was an honorary member of the Institute for Tropical Medicine in La Habana. He trained in Italy, and he was very honored to have been made a honorary member of the Italian Society of Parasitology. And one of the labs at Dr. Oswaldo Cruz Institute, renowned institute in Brazil, bears his name, and he was recognized with the post of president of the World Federation of Parasitologists. In the meantime, cases were being described in other countries, predominantly in Latin America, but eventually even in the United States. 2 cases in urban Los Angeles in 2 kids that had did not have any travel history. So this is considered the first autochthonous infestation in United States. And 50 years later, we've learned a lot more about this parasite. We, for example, know that it's not associated with socioeconomical status or malnutrition, kind of underscoring the accidental nature of the contact in humans. We have a growing but still quite incomplete knowledge of its molecular biology. We know that medical treatment is actually not very effective and actually could be problematic because it may create a larva migrans type of picture. And most of the preventive measures for this disease have focused on trying to control the intermediate and the definitive host, including mandatory cleaning of vegetables in Costa Rica with 1.5 chloride solution. Of note, refrigeration, is not enough to kill the larvae. It is considered endemic in Costa Rica, but unfortunately, take a look at this report. Almost 90% of the cases have been described in Costa Rica. There's absolutely nothing special in the environment in Costa Rica, whether this parasite would only grow there. So it's very likely that the problem is that there is subdiagnosis and sub-reporting in the rest of the countries. So in conclusion, I think this is a story of whether a serendipitous finding recognized by careful observation and intellectual curiosity, where close collaboration between different fields in medicine came up with solving a problem that also required a lot of rigorous scientific investigation and kind of keeping at the problem and trying to go deeper and deeper to try to find answers. And in this case, their knowledge and their findings did have a significant impact in public health and medical treatment in a population that is quite vulnerable. And I just want to end with giving thanks to Dr. Maximiliano Rodriguez and Dr. Fernando Brenes, who helped me with some of the material that I presented, and especially to Dr. Silvia Molina. She was my parasitology teacher in medical school, and was the first person from whom I heard this story. And 3 decades later, I'm still fascinated by it. Thank you very much.
Any questions or comments from the audience?
Just a naive question. Why do you think the cases bulk in children and young adults?
That's a good question. Most likely in kids, it's likely because they're playing around, they can eat, actually it's more likely that I actually eat the mollusk itself, as disgusting as that sounds, than just the secretions of the mollusk. But, just by playing around in the garden or in the yard are more likely to acquire the disease.
Another
naive question. How is the diagnosis made nowadays?
Still, histological confirmation is the gold standard for the diagnosis. The ELISA is not 100% specific because it cross-reacts with infections of Angiostrongylus cantonensis and even other species of Angiostrongylus that are not pathogenic. But on the right clinical setting, with a positive test is considered quite suspicious, and patients usually go to surgery and have removed. It's not, like I said, it's not clear that just medical treatment is effective or sufficient.
I have a question,
Yep.
if it's time. Are there any theories to explain the particular anatomical tropism of this parasite?
No, and it's likely related to the molecular biology of the parasite. Of note, the Angiostrongylus cantonensis, what it provides is a similar picture, but in the brain. So the cantonensis tend to migrate to the vessels at the base of the brain, at the base of the skull and the brain. Costaricensis tends to go to the mesenteric sites. And it's likely has to be mediated by protein expression at those tissues, but it has not been identified which is the specific cause.
Thank you.
I was also very surprised that there's such a high prevalence in Costa Rica in contrast to the countries that are nearby. Do you think they just don't see the parasite, or they named it something else?
No, I think it's a combination of not appropriately diagnosed and most likely not reported in a systematic way.
Okay. And are you that young that you never cross paths with him? In your
Not that I know, but I haven't done the ELISA assay. So.
No, but have you crossed paths with Dr. Herrera?
Yes, and my last year of medical school, or maybe my internship, I don't remember. I remember attending to Dr. Céspedes' retirement session.
Also, that's great. Thank you.
Thank you.
Rafael. Thank you.